Hello students, welcome in today's lecture. In previous lecture we have started our topic, the molecular basis of inheritance, and in that we have seen DNA as a genetic material, and in this point the first experiment that we have seen is the Griffith experiment, who proved that the transforming principle is there which convert the live R strain bacteria into the S strain bacteria. Now the second experiment that today we are going to discuss is the experiment which is carried out by three scientists that are the Avery, MacLeod and McCarthy, who also prove that DNA is only the genetic material which can pass from one generation to the next generation. So second experiment that we are going to discuss here, that is Avery, MacLeod and McCarthy experiment. These are the three scientists, Avery, MacLeod and McCarthy. These are the three scientists who carried out the experiment in 1944. After the Griffith experiment, he carried out the experiment on the mice, and what they did, they purified the biochemicals from the dead S strain bacteria. They purified the biochemicals from the dead S strain bacteria. And which biochemical they purified or isolated from that S strain bacteria? These biochemicals are the protein, DNA and RNA. These three biochemicals they isolated from this dead S strain mice, and these biochemicals are the protein, DNA and the RNA. And what they did, they have to prove that which factor or which biochemical transform the live R strain bacteria into the S strain bacteria, and for that purpose they isolated these biochemicals from that dead mice or from the S strain of the bacteria, and they prove that only the DNA from this biomolecule can transform the R strain bacteria into the harmful virulent S strain bacteria. They found that protein and RNA do not have any role in the transformation of R strain into the S strain. Only this DNA is responsible for transformation of R strain into the S strain. They also discovered some enzymes. That means they discovered enzymes like the RNAs. They discovered an enzyme like protease. They discovered the enzyme like DNase. They also found that these enzymes, RNase means the enzyme which digests the RNA molecule. Protease means the enzyme which digests the protein molecule, while DNase means the enzyme which digests the DNA molecule. So they also discovered these different enzymes, and by these enzymes they also tried to prove that which material is responsible for conversion of the R strain into the S strain, and at the same time they found that these RNAs and protease also do not inhibit transformation, but the DNase enzyme is only enzyme which inhibit the transformation. Now I will explain how this DNase enzyme is responsible for inhibition of the transformation. We'll see the role of this DNase enzyme. What happened here? That means Avery, MacLeod and McCarthy took a test tube. In that test tube, they placed they kept this R strain. At the same time, in that same test tube, he pour the DNA from the dead mice that they were isolated, and they pour this DNA into this test tube which has the live R strain. They found that all the R strain in this test tube gets converted into S strain, because this R strain pick up the DNA of the dead mice of the S strain, and due to which all the R strain, due to the transmission of the DNA into R strain, gets converted into the S strain. At the same time, what happened? In the next test tube, they again pour this R strain. At the same time, they pour DNA from the dead mice, and in the same test tube, they also added the enzyme DNase. As I told you that the role of this DNase enzyme is that it digests the DNA. When this DNA is kept in this test tube, at the same time DNase enzyme is also added in the test tube. What happens? This DNase enzyme digests this DNA which is released in that test tube, or which is placed in that test tube from the dead mice. And due to what happened? This DNA, due to the digestion of this DNA enzyme in the test tube, is not taken by this R strain, and that's why when we analyze the result, what you will find that in the test tube R strain remains as it is. They do not get converted into the S strain, because this DNase enzyme digests this DNA, due to which the DNA is not taken inside the R strain, and R strain cannot converted into the S strain. They remain as it is, the R strain. And this experiment also proves that the DNase enzymes also have the role in the inhibition of the transmission, because DNA is only responsible for their transformation R to S, and when that DNase enzyme get added with the DNA, then definitely this DNase enzyme digests the DNA. मी तुम्हाला एकदा सोप्या भाषेमध्ये मराठीमध्ये सांगते, म्हणजे तुमच्या लक्षात येईल व्यवस्थित. काय सांगितले बघा आपण इथं, Griffith's experiment same repeated by 3 scientists, Avery, McLeod and McCarthy. They found that, काय त्यांच्या लक्षात आलं पहा, they found that the R strain which we have seen in our previous lecture gets converted into the S strain. एवढं तर तुमच्या सगळ्यांच्या लक्षात आलं का बघा. ग्रिफिथच्या एक्सपेरिमेंट मध्ये आपण काय बघितलं, आर स्ट्रेनचं कन्व्हर्शन एस स्ट्रेन मध्ये झालं, कारण काहीतरी असं आर स्ट्रेननं एस स्ट्रेन मधून घेतलं आणि त्यामुळं काय झालं, सगळे लाईव्ह आर स्ट्रेन एस स्ट्रेन मध्ये कन्वर्ट झाले. हे आधीच कळलं असेल तरच हे व्यवस्थित समजेल तुम्हाला. त्यामुळे इथून आपण स्टार्ट करतोय, आर स्ट्रेन गेट कन्वर्टेड इनटू एस स्ट्रेन बिकॉज these R strain have something accepted, and due to which this R strain get converted into the S strain. But which that chemical accepted by this R strain, that tried to find out by these 3 scientists, Avery, McLeod and McCarthy. That's why what they did, they carried out their experiment in a test tube. They collected different type of test tube. In each test tube they added, for example, suppose they had taken the 3 test tube. In one test tube they had added the R strain, in second test tube also R strain, in third test tube also R strain. Means 3 test tube carry the R strain, which is the non-virulent one. Okay. At the same time, in first test tube he added protein from the dead mice. In the second test tube he added RNA from the dead mice. In the third test tube he added the DNA from the dead mice. And surprisingly, they found that in the third test tube, in which they added the DNA of the dead mice, all the R strain get converted into the S strain. But the first 2 test tube, in which protein and the RNA added from the dead mice, are not converted into the S strain. And 100% they here concluded that only this DNA is responsible in a third test tube which convert the R strain into the S strain, and thus they prove that DNA is a transforming principle which accept by R strain and convert into the S strain. Again, for their confirmation, what they did in the last test where they added the DNA of the dead mice in R strain, at the same time they added the DNase enzyme. And what happened? First, this DNA enzyme digests the DNA, and due to which what happens, this DNA get digested and not accepted by the R strain, and thus R strain remain as it is, they not converted into the S strain. It is very simple experiment. You have to read it repeatedly from the textbook, and if you have any doubt about it, then you call me personally. I will again explain you. Okay, this is about the every MacLeod and McCarthy experiment, which experiment is not accepted all the biologists, and so they cannot prove that the transforming principle is the DNA. But if there is question asked for the NEET exam that who proved the transforming principle is a DNA, then the answer of that question is that every MacLeod and McCarthy proved that transforming principle which was discovered by Griffith is definitely DNA. But their theory is not accepted all over the world. So third experiment which proved that DNA is the genetic material, and it is proved by the scientist Hershey and Chase. So the last experiment under the title DNA as a genetic material, that is a Hershey and Chase experiment. Now we are going to study by this diagram. So second experiment we have completed here, that is every MacLeod and McCarthy experiment. Then what is the third experiment that we are to see here? That experiment's name is Hershey and the Chase experiment. So this diagram is drawn here on the board, and this diagram proves that how this Hershey and Chase carried out his experiment. So last and third experiment that here we are going to discuss, that is Hershey-Chase experiment. If there is question asked for MCQ that who proved the DNA as a genetic material, then the answer of that question is that Hershey and Chase proved the DNA as a genetic material, because in their experiment they definitely show that DNA can transfer from one generation to the next generation. And which was their experiment, that we are going to discuss here. See here, for their experiment they carry their experiment on the bacteriophage. Bacteriophage means the virus which infect the bacteria, that we call as a bacteriophage. For their experiment they carried out their experiment on the bacteriophages, and as we know, these bacteriophages are made up of two important components, and those components are DNA and the protein. DNA and the protein, these are the two important components which made this bacteriophage, and that bacteriophage were carried for their experiment by this Hershey and Chase. What happened next in their experiment? They grow some of these bacteriophage, that we simply say the viruses. Here we have seen the structure of this bacteriophage in the last year in 11th standard. You know very well the structure of the bacteriophage. These are the diagrams of the bacteriophage. We simply say here the viruses. Okay, he has taken the two viruses. Some viruses he has grown in a medium, which medium contain the sulfur, and so he labeled that bacteriophage which grows in a medium, that medium contains the sulfur, and sulfur is of the, he labeled that bacteriophage or the viruses which grow in the sulfur medium as the 35S bacteriophage. While some viruses he grow in another medium, that medium contain the phosphorus, and so he labeled that bacteria as the 32P. P stand phosphorus, S stand for sulfur. What I said here, see here, the two viruses he collected. Some viruses he has grown in a medium, that medium contain radioactive labeled sulfur, while some medium contain the radioactive label phosphorus. So two mediums are there. One medium contains the sulfur, one medium contains the phosphorus, and he has grown these two viruses in these two different medium. Some viruses grown in the phosphorus containing medium, some viruses grown in a sulfur containing medium. Then what happened next? We know the property of bacteriophage, that any bacteriophage do not enter into the body of the bacteria. So this is the structure of the bacteria, and if any bacteriophage attaches to the surface of bacteria, he first inject its genetic material into this bacteria. So in next figure, what you will see here, this bacteriophage which attach on the outer surface of the body of bacteria injects its genetic material inside the bacterial cell, but the whole body of bacteriophage remains outside this bacterial cell. See here also same, this bacteriophage injects its genetic material inside the body of bacteria, but bacteriophage do not enter into the body of bacteria, it remains outside. So these viruses attaches at a special surface on the body of this bacteria and insert their genetic material only into the bacterial cell. So the second figure is, the name of this figure is we call as an infection, and to avoid the space, second title is also given to same figure, that is the blending. Then what is meant by infection and what is meant by blending? First step in his experiment, the name is given as the infection. Infection means when any bacteriophage infect the bacteria, inject its genetic material inside the bacteria, that process we call as an infection. Blending is the second step. What is meant by blending? Blending means that genetic material of the virus mix within the genetic material of the bacteria, and that process is called as the blending. To avoid the space, again I am saying that I have reduced one figure. So this figure is given as the name infection as well as blending. That means here you will see that the genetic material of the virus and genetic material of the bacteria, genetic material of virus and genetic material of bacteria mix to each other, and that process we call it as a blending. And third one is process that is given here, which is called as a centrifugation. What is meant by centrifugation? Centrifugation means it is a technique in which there is the application of the centrifugal force, due to which the particles from the solution get separated according to their size, shape, density, viscosity, etc. And same technique is applied here. Here what happens? These bacterial cells are centrifuged, and due to which the particles which are present inside that bacterial cell get centrifuged, and due to the centrifugation property, the result found is that in this medium where the viruses are grown in a phosphorus containing medium, show that phosphorus detected in cell and no phosphorus detected in supernatant. Means after centrifugation, what happens? After centrifugation, there is a liquid content present above that solid material, and in that liquid content you will find that phosphorus is not detected, because all the phosphorus are present in the virus, and so phosphorus is detected in the cell. In the next figure, what happened? The bacteriophage which is grown in the sulfur medium, where after centrifugation sulfur is not found in the bacterial cell, but that sulfur you will get in the supernatant. That means sulfur remain in that liquid portion, it is not enter inside the bacterial cell. One conclusion is very important that I am going to repeat here, and then you will understand this experiment very clearly. Again I will repeat the experiment, that Hershey and Chase for this experiment has taken two bacteriophages. These bacteriophages are made up of the DNA and the protein. That means the chemical composition of the bacteriophage is that DNA and the protein. These bacteriophage he has grown in a different medium. One bacteriophage he has grown in a phosphorus medium. That phosphorus is labeled as 32 radioactive phosphorus, while some viruses he has grown in a medium that is labeled as a sulfur, that is the 35 S sulfur level medium is there, which is a radioactive. Why it is radioactive? If you have to see which material enter in the bacterial cell, then radioactivity is very essential. That's why it is labeled by the radioactive. Then what happened? He has also taken the E. coli bacteria. So these bacteria are the essential coli bacteria. After the growing of that virus in a phosphorus and sulfur medium, he allowed this bacteria to infect the E. coli. He allowed these viruses to infect the E. coli. So here you will see that the viruses which grow in a phosphorus label medium, these viruses get infected to the bacteria. After infection, these viruses added their genetic material in the bacterial cell, and that process we call as a infection. After adding the genetic material in a bacterial cell, that genetic material of the virus cell get mixed with the bacterial cell. That process we call as a blending. And the third step is that here this bacterial cell which has the genetic material of its own as well as genetic material of viruses undergoes the process of centrifugation. In centrifugal force, there is a separation of the particles of the bacterial cell. There will be supernatant in that centrifugation technique. Supernatant means liquid medium is present above the solid medium, and here you will find that in that supernatant and in that liquid which is present above the solid, there is no phosphorus present, because all the phosphorus which was present in that viruses, after infection to bacterial cell, he injects that phosphorus into the bacterial cell in the form of DNA. Because if we see the chemical composition of DNA, this DNA is made up of phosphorus. So all the phosphorus taken in that viruses get injected along with its DNA into the bacterial cell. So only that phosphorus you will find here in the DNA of its genetic material, but it is not found in the supernatant. But in the next figure you will see that the bacteriophage which were grown in a sulfur medium, when they are allowed to infect the E. coli bacteria, they do not inject this sulfur into this bacteria, because sulfur is not a component of the DNA. Sulfur is a component of the protein, and that's why you will not find this sulfur in the bacterial cell, because sulfur is not a part of DNA. And here in the supernatant you will find the sulfur, because all the sulfurs you will find in that liquid portion, it is not injected into the bacterial cell. And thus this experiment proved that viruses has one of the important property, that it always inject its genetic material DNA into the bacterial cell. So as the phosphorus is a component of DNA, all the phosphorus goes into this DNA, and along with DNA that phosphorus enter into the bacterial body. But sulfur is not a component of DNA, so sulfur do not enter into the DNA of the bacteriophage, and with this DNA, that sulfur also do not enter into the bacterial cell. So that sulfur is not found in the bacterial body, but it is found in the supernatant. Phosphorus is found in the bacterial body, but it is not found in the supernatant of that centrifugation. So it is very simple one, that only one sentence proves this experiment, that DNA is the genetic material which get transferred from bacteriophage to the bacteria, and along that DNA phosphorus also transfer, but sulfur cannot transfer, and this experiment proves that DNA is the genetic material. Again, I think that if you do not understand the same experiment, again you also call me, I will explain individually, because these experiments are very easy, but they have given the different steps, that's why some students can't understand at first level. So this is the experiment of the Hershey and Chase, and this experiment also proves that DNA is a genetic material. So here the three experiments we have seen. First one is the Griffith experiment, who told that the transforming principle is there which convert the R strain into the S strain. Second experiment, that is of the Avery, MacLeod and McCarthy, who also prove that the DNA is a transforming principle, but their experiment was not accepted all over the world. And the third scientist, Hershey and Chase, they carried out the experiment on the bacteriophage, and they proved that DNA is the hereditary material which can pass from one generation to next generation, or which can pass from one organism to the another organism, and thus their experiment was accepted all over the world, and thus there is final proof that DNA is a genetic material which was given by scientist Hershey and Chase. The number of questions are asked for the MCQ, that is, who proved the DNA as a genetic material, then the name is given by the Hershey and Chase. Also there are questions asked that which phosphorus medium is used, that is we call as a 32P, which sulfur medium is used, 35S, which the experimental material is taken by Hershey and Chase, this bacteriophage. And now the next point to this is the packaging of DNA. That means the DNA we have seen as a genetic material, how that DNA get packed in the nucleus of the cell, because this DNA has a long length, about 2.2 meter is long, and such a long DNA in a very small nucleus can get packed at a very different level, and that we have to see in the next point, and that is called as the packaging of the DNA. So in today's lecture we have completed all the three experiments: Griffith experiment, Avery McLeod-McCarthy experiment, and Hershey and Chase experiment, under the title DNA as a genetic material. The three experiments are very important. Read it carefully and you will find that there is a little difference between the three experiment and which prove the DNA as a genetic material. Thank you.